Very good day to everybody present here. So in this particular class on information technology in bioinformatics and genetics, we would be discussing about comparative genomics. So this is the pre-ultimate topic of the bioinformatics and genetics. So in comparative genomics, we will discuss about the formation of orthologs and paralogs. Then we discuss about the comparative genomics of bacteria. Okay. So this is especially for you, my dear students and young researchers, and you can reach me at dr.christoananth@gmail.com. So before beginning the session, let me thank God for giving me this golden opportunity to deliver this useful session, to share my knowledge among my fellow national, international participants, students, and young researchers. In today's session, we'll have a short introduction or intro to comparative genomics. Then we understand what are the driving forces for comparative genetics, and then we discuss about biochemical activities, about the protein evolution which occurs by the exon shuffling. Then we understand how that's a particular example, how hemostatic proteins can be considered as an example for the exon shuffling. Then we discuss about the comparative genomics of bacteria. Then we determine the minimal gene set which is consistent with the independent existence. Then we try to form COGs, clusters of orthologous groups. Okay. So finally, we are going to end this class with an open-ended project. I've already given them in the Google Classroom. Please complete them at your best available time. And at regular interval, we'll have some video to discuss the knowledge in our topics. So whenever you are going to discuss about comparative genomics, we are going to understand the study of the differences, the differences as well as the similar features in this genome structure. And of course, we are going to utilize these phenomena in the organization of the different organisms. So maybe that would be a question that would be arising. So what about the differences between the humans and the other organisms which are reflected in our genomes? And also how similar would be the number as well as the type of the proteins in human beings or maybe fruits or maybe flies or maybe even small worms, plants, yeast as well as bacteria. So maybe this comparative genomics is nothing but the application of the bioinformatics methods, and this is being applied to the whole of the genome sequences. And definitely generally we tend to apply this biology in silico principle as well, and we tend to take this as a very extremely powerful technique, and generally you can provide much more biological insights can be achieved with the help of this comparative genomics. So these are the driving forces, the useful phenomena for this comparative genetics or genomics. So we should have, the a thorough understanding of the process of the evolution. Then only then we can able to characterize these phenomena in the local level. Okay, so maybe you can make this related species much more unique or peculiar even. And generally, there is another phenomena which is driving for the comparative genetics, which is nothing but translating the DNA sequence data into proteins. So that's what you try to go with the comparative genetics. And definitely this DNA sequences generally you should be conserved, it should be protected between the species rather than the sequence encoding. So maybe if you consider for the non-coding sequences or maybe coding with dispensable functions even, it has to be protected at most. So generally you can consider these comparison for comparative genetics, maybe in the physiology or maybe behavior of structures even, and even for the non-conventional or maybe non-functional sequences also you tend to apply this comparative genetics. So this is where we tend to form orthologs or maybe paralogs. For example, if you are going to compare with the mammals or maybe fish even, you will have to identify conserved sequences even. So everything has a specific or maybe significant function that are associated. So here we would have homologous genes which are nothing but orthologs. So you are going to encode the proteins with the same function. So that's what you generally evaluate by this direct vertical descent method. But if you take in the case of encoding proteins, with related but non-identical functions. You can use paralogs as maybe homologous genes. Generally, you can encode proteins with the relative but non-identical functions. So you can summarize that orthologs generally evaluate maybe evolved from this gradual accumulation of mutations, but paralogs it will be arising from the gene duplication. Okay, so we'll have gene duplication and then we'll have mutation accumulation. Okay. So even with the case of different examples, we'll have protein superfamilies, which are considered as better examples for paralogs. And there are several biochemical activities that are taking place in the living organisms even. We have citric acid cycle, we have ATP, that is nothing but adenosine triphosphate, synthesis of nucleotides, DNA replication, and so on. So generally, if you consider the key proteins would be this orthologs, as we discussed. The protein superfamilies are paralogs, and key proteins are nothing but orthologs. So here, if you consider the universal protein families, maybe you'll have RK, eubacteria, eukaryotes. So they are considered to be the universal protein families. Okay. So generally, there's increasing evidence even whether these functional equivalents of the proteins require sequence similarity and not even three-dimensional folds even. Okay, so that's the way it goes. So whenever you're considering orthologs, it's responsible for the same function in different organisms. So what you call to be the non-orthologous gene displacement. And protein evolution occurs by exon shuffling event. So maybe if you are going to sequence maybe close to 200 different genomes, so maybe you can see that this gene displacement occurs within most essential genes. Okay, so even at least two biochemical solutions to each cellular requirement would be there, and only about 60 genes can be identified where you consider this gene displacement has not been observed. So, most of these would be trying to encode the components of the transcription and translation systems. And it has been found that this analysis of protein sequences and 3D structures has been revealed. So maybe it is composing of discrete domains even. So you call these to be the mosaic patterns, and these are much more prevalent in the metazoa. And majority of the mosaic patterns are extracellular and not intracellular. Okay, so and they would be since they are extracellular, they would be constituting only extracellular parts of the membrane bound proteins, and definitely it plays a very important role in the multicellularity. Okay, so maybe the individual domains of this mosaic protein is involved in the specific functions, and definitely since they are extracellular, they are evolutionary mobile in nature. They would be spreading during this evolution, and it occurs in the unrelated proteins as well. So you can say that this mobile domains are characterized by this, its feature to able to fold independently. Okay, so even it's considered as a very good important characteristics because it is going to prevent the misfolding feature. That's a problem when they are inserted into this new protein environment even. So maybe up to date we have found like 60 mobile domains as well. So this is exon shuffling. So we have taken a survey of genes that is going to encode the mosaic proteins. So it will be experiencing a very strong correlation between this domain organization and the intron-extron structure even. So generally each domain will be encoded to one or maybe different combination of exons even, and new combination of exons are actually created by this recombination with the intervening sequences as well. Okay, so this process, what you have to be the rearrange genes with altered function, you call it to be the exon shuffling. And because this average intron is larger than the average exon, so the recombination frequency is proportional to this DNA length. Okay, so there are many crossovers which occurs in the non-coding sequences as well, and this repetitive sequences will give rise to this exon shuffling by promoting this mismatching and of course the recombination of the non-homologous genes. So it's all about this recombination which drives the entire exon shuffling process. So you consider hemostatic proteins as a good example of exon shuffling. So here, whenever we have like blood coagulation or maybe fibrinolysis, so it will be evolving, enzymatic reactions as well. So in which inactive zymogens are converted into active enzymes, and this zymogens generally it will be having, belonging to the family of serine proteases, and this activation will be having this proteolysis of a limited number of peptide bonds. And here, when you try to compare this amino acid sequences with this archetypal serine proteases, maybe you take in the case of trypsin, maybe you will have, this amino acid sequences would be having large N-terminal extensions. Okay, so these extensions we will have different number of discrete domains. We will be having some functions called substrate recognition, binding of cofactors even. So definitely there will be a very strong correlationship with the exon structure. So that's where you tend to get this exon shuffling out of this hemostatic proteins. And we will have microbial genomes as well. So even though if we have observed in the previous slides, like the mosaic patterns are much common in the metazoa, even in the unicellular organisms also we would obtain these mosaic patterns. Okay, so large number of this microbial genomes have been sequenced, and we will have archaea, eubacteria, eukarya, so that is considered to be the universal protein families. So maybe you can actually determine very easily the evolutionary mobility of the domains. So maybe if you can observe this genomes of 15 bacteria, 4 archaea, 1 eukaryote, generally it will be consisting of domains of different genomes because it's considered as a universal protein families. Okay. Also, they have found like 37 examples of proteins which consist of a native domain, and maybe there is a horizontally aligned, alien domain as well. Okay. So in several of the sequences, the genome would be containing the gene for the mosaic pattern as well as the sequence encoding. So, only thing you are able to witness this universal relationship. So this is the comparative genomic of bacteria. We'll have bacterial DNA and then the plasmids. So the genome, if you can consider the size, it will be varying from 0.49 megabyte. Okay, so that is the case of the nano archaeum equidens to nearly 9.1 MB. Okay, that is in the case of the Brady Rhizobium japonicum and two species of the streptomyces. Okay, so that's actually, if you compare like 0.49 and 9.1, it is like 18 times it is having the difference. Okay. Also, the gene density is similar across all the species, and generally it is about like one gene per kilobase of this DNA. So you can observe that there are larger prokaryotic genomes as well, which is containing, once again it is consisting of more genes than the smaller ones. Okay, so there are larger prokaryotic genes. Also, when you consider this archaeal genome, so it will be having like a different smaller range of genome sizes because it's small. Okay, so it can be considered as an artifact of the small number of genomes. And whenever you are considering this bacterium, it will be generally in different range of environments it will be living, and definitely the maximum size will be 5.8 MB. Okay, so that is considered as the largest archaeal genome. Smallest means you can find it in maybe animals or maybe human beings, maybe in the form of mycoplasmas or maybe rickettsias or maybe chlamydia. Okay, so that is how it goes. Big, it is found to have a size of 5.8 MB. Small, you can find it in the human beings like mycoplasmas, rickettsias, and chlamydia. Okay, chlamydia. And we tend to determine this minimal gene set which is consistent with the independent existence. So generally it will be having these organisms will be having larger genome size, and you can establish a very good correlation between the genome size and the metabolic. So you can find it in this, as I told you, streptomyces or maybe rhizobia or maybe pseudomonas even, where you can have degradation of wide range of aromatic compounds. And generally, this genome of n equidens will be considered as the smallest sequence to date. But definitely, you will have observed minimal set of genes as well, which we would be having most favorable conditions in which all the necessary nutrients are actually provided, and there is no stress factors, no environmental stress factors even, and even you should consider that this minimal protein set is considered to be the problem and you have to solve it. So this is the minimal protein set. So we'll have a list of orthologous proteins in the Haemophilus influenza, what you call to be M genitalium, and generally you will have like proteins for cell survival, and as both bacteria and it should have composite of the survival genes. Okay, so altogether you can find out like 244 orthologs have been identified, and it's very unlikely to complete, okay, because of the occurrence of this non orthologous gene displacements. So but some of these gene displacements can be inferred because both organisms will be having key metabolic pathways that are incomplete, and generally you will have minimal protein set which is extended to almost 256 genes. So that's the criteria that you can extend with the minimal proteins. And also we tend to form COGs. So even with the case of minimal protein set, that's taken to be underestimated factor, and variation is to identify this orthologous groups, and we have cluster of genes which will include orthologs, and definitely we'll have paralogs even, so we'll have gene duplication, and maybe you'll have maybe 4 eubacteria, 1 archaebacterium, and at least 1 yeast even, so we will be forming COGs, clusters of orthologous groups. We will be forming 816 clusters of orthologous groups which were identified, and of these 327 contain the representatives of the three kingdoms. Okay, so based on this set of 327 proteins, it was possible to reconstruct all the key biosynthetic pathways as well. And whenever you are trying to repeat this analysis with the sequence data, so you'll have 3 archaebacteria, 12 EU bacteria, and maybe you'll find this minimal protein set would be reduced to 322 COGs, 322 clusters of orthologous groups as well. So this is the open-ended project work. I've already given them in the Google Classroom. Please complete them. Please complete them at your best available time, and I will be checking them, and wish you all the best for the forthcoming session.